Hello everyone and welcome back to our tutorial series on analysis of biomolecules. Today we will look into the chromatographic mode of hydrophobic interaction separations and how it is used to analyze proteins. First of all, a very short repetition of what hydrophobic interaction chromatography actually means. So what we will do by that is separate molecules by their hydrophobicity. Hydrophobicity is a word that contains two parts, one of which is hydro. You very likely know it is relating to water, and phobia, something which means fear. And the molecules we will talk about are actually molecules that are basically afraid of water or don't feel as comfortable in polar conditions. And what we will do is to separate them by how much they don't like water, basically. So how can we now achieve the separation of differently hydrophobic molecules? So what we use is a stationary phase that bears a ligand that is hydrophobic. So typically a butyl phenyl functional group that is on our stationary phase. And then we load or bind our hydrophobic molecules in a high salt concentrated buffer. And this high salt concentration leads to the fact that we have a low solvation of the molecules and we have a very good accessibility of the hydrophobic parts on our analyte molecules and also on our stationary phase. Now, if we reduce the salt concentration, what will happen is that we get back the solvation of our molecule, so we don't have this accessibility of our hydrophobic parts on our analytes. And finally, if we go very low with the salt concentration, they will elute. Well, here we have now even more visual representation of how hydrophilic interaction chromatography works, as the salt is basically displayed. So for binding, we have the high salt concentration, we have a lot of ions, and you can imagine that the ions in combination with the dipole of the water keeps the water very busy here, and not a lot of free water molecules are there to build up or to solvate our stationary phase or molecule of interest, so that we have this accessibility and the interaction between the hydrophobic parts of stationary phase and our molecule of interest. Now, if we take away all of those salt ions, we in contrast have a lot of water there, which can solvate our molecules. We don't have the hydrophobic parts accessible anymore, which means we have elution from our column and the separation by hydrophobicity, meaning the less hydrophobic molecules will elute earlier than the ones that are more hydrophobic. Now that we know how hydrophobic interaction chromatography works, one of you might think, okay, but there are two methods that separate our molecules by hydrophobicity, so why should I actually take the hassle of doing HIC, whereas I'm much more familiar with reversed phase, for instance? If we look into the comparison of both, what we see is that for HIC we use a salt gradient, whereas we use organic solvents for reversed phase mobile phases. And this actually leads to the important and last point in this table, which is protein denaturation. So if we use organic solvents, this means we will denature our proteins, so basically also the secondary structures of our proteins, which is not the case if we use salt aqueous buffers. This might not be as 100% true as it's written here, so there are indications that we might have a little unfolding on the column if we use hydrophobic interaction chromatography and expose our proteins to very high salt concentrations, but it's not as drastic as for reversed phase separations. Other differences is the compatibility with mass spectrometry. There definitely we have an advantage for reversed phase applications, because you can directly connect your separation with mass spectrometry, which is not the case for hydrophobic interaction chromatography, of course, as there we have a high salt concentration, which is not compatible. Last but not least, if you think of the method development for hydrophobic interaction chromatography, it's often regarded as very tedious. There are some things we need to take care of, and sometimes it's rather a try and error and not really, okay, I know I have this problem, I need to change parameter A and then I'm fine. So we have to think of the salt type, the salt concentration, the gradient that we use, and last but not least, we don't have so much literature and expertise out there for hydrophobic interaction chromatography as compared to reversed phase, where there's a lot of information readily available. So why is it actually possible to separate proteins by hydrophobic interaction chromatography? What we find on proteins is that their surface contains parts that are more hydrophilic and parts that are more hydrophobic. And these hydrophobic parts, or hydrophobic patches as we call them, they interact with the stationary phase and are responsible for retention. Furthermore, there are many modifications we can find on proteins and their amino acid side chains that have an impact on the hydrophobicity and how our molecules elute from the column. So we have the possibility to look at tiny changes, modifications in our molecule by hydrophobic interaction chromatography. Just to name a few, one of them is deamidation, for instance, from an asparagine to aspartic acid, where we have a decrease in the hydrophobicity. If we look at methionine oxidation, we also will find a decrease in hydrophobicity, whereas if we increase the size of our molecules, we get an increased hydrophobicity, and if we attach any modifications that are more hydrophobic than our protein itself, of course this will also lead to a hydrophobicity, and this is often done, for instance, for antibody drug conjugates, which use a hydrophobic chemotherapeutic drug attached to an antibody. And talking about ADCs, we already entered the part of what can actually be done with using hydrophobic interaction chromatography. So one very typical application of hydrophobic interaction chromatography is to analyze the drug-antibody ratio of antibody-drug conjugates. So as I said, antibody-drug conjugates, they carry hydrophobic drugs on there. So what will happen if this is in the body? The mAb is basically responsible to get the drug to the site where it should act, basically, maybe in a tumor cell, and then the chemotherapeutic is released and directly acts where it's needed, meaning in the tumor tissue. But of course you can imagine it's very important to know how much of these chemotherapeutic drugs are attached to one antibody, as this is the actual drug that works afterwards. And for this, hydrophobic interaction chromatography is used, and what we see is that the antibody without any modification elutes first. Of course, it does not have any hydrophobic modification on there. And then we get later elution of the antibodies that carry either 2 or 4 or even more drugs on there. And from there you can get back also to the number, or to the mean number of drugs attached to an antibody, which is the drug-antibody ratio. Now, one thing that we saw on the overview of things we might be able to separate by Higgs separations is deamidations. So deamidations occur during storage, aging of proteins, and this means we get from an aspartate to an asparagine or isoaspartate. In order to look at if we can separate those modifications by Higgs, what we did is we took an antibody sample that contained some of those modifications and we fractionated it on ion exchange columns. So what we got is samples that contain either the modification on the heavy chain of the antibody. We have a fraction that does not contain any modification on aspartates, and we have a sample where an aspartate on our light chain was modified. Of those fractions, they were enzymatically digested in order to get the different parts of an antibody, meaning we have a cleavage into the Fc part and the two Fab parts. Now, if we run those fractions and digested antibodies on a Higgs column, what we will find is, on the one hand, shown here on top, if we have our modification on the heavy chain, we will have two peaks where we expect our Fab part to elute, and the earlier one being less hydrophobic and the modification into an isoaspartate. Of course, we also have our non-modified sample, and there we basically have just one peak that elutes where we expect our Fab parts to elute. And if we go into the last fraction here, what we will see is again two peaks, as one of our Fab parts has a modified aspartate to asparagine in the light chain. Just like deamidations, also methionine oxidations are modifications that can occur on a protein, for instance, due to long or wrong storage, and actually are a first sign of degradation of the protein. So what happens is, of course, as the name says, we have our methionine where the sulfur is oxidated, and what we did in order to see if we can detect it via hydrophobic interaction chromatography is to expose an antibody to increasing percentages of an oxidative reagent. And what we will see or see best here in the top green chromatogram that was exposed to the highest concentration of the oxidative reagent is that for the Fc peak, we will find not only one peak, but actually three. And in order to explain this, we need to know that our Fc part actually contains two methionines, and therefore we have two possibilities to oxidate and probably have those three peaks. Again, we find the peaks smaller and eluting before our main peak, which indicates that we have a less hydrophobic structure in here. I promised in the very beginning that Higgs is actually a very versatile method, and as we were just looking at the very small modifications that can even be dedicated with hydrophobic interaction chromatography, there are also bigger changes that can be looked at when using hydrophobic interaction chromatography. So what we are looking at here is a publication from last year, where via Fab arm exchange, so Fabs are these arms of an antibody, and addition of nanobodies, a multispecific or actually for specificity antibody was generated. But of course it's super important to know, okay, do I have really a Fab arm exchange, meaning I have two antibodies, but what I actually want in order to get two specificities is that in the middle. So we need to know, is it composed the right way or not? And as in this case, the parental antibodies were actually very different in their hydrophobicity. If we look at this and this, we do see that this gray one is much less hydrophobic than compared to this brighter one. And if we now have the Fab arm exchanged, what we will find is something in the middle. So also for these kinds of things, it's possible to use hydrophobic interaction chromatography. And I would like to line out one more example of how hydrophobic interaction chromatography is. And this is very simple actually. It's just to know the rough hydrophobicity of a molecule, and this is very important as a molecule or an antibody, their hydrophobicity impacts on how prone they are to aggregate, how well they will be soluble, and how well the molecule will be cleared from the body. So all physiological effects that you can already look at a very early stage of your drug development, and it's actually very important to do so, because otherwise you would continue with molecules that are much too hydrophobic to at the end have good performance in the body basically. And what you do is just to screen your different proteins or protein candidates, run them on a hydrophobic interaction chromatography column, see when they elute, and from there you can get back to the hydrophobicity or surface hydrophobicity of your molecule and decide, is it okay to move on with the molecule or not. Well, now I hope every one of you found at least one application that might be helpful where they can use hydrophobic interaction chromatography. And you might think, okay, in the very beginnings I said it's regarded very complicated to do the method development. What does method development for hydrophobic interaction chromatography actually mean? So we do not have too many parameters we need to look at. One thing is of course the columns. Then the salt type and the concentration is a very important factor that we can use to fine-tune our method. pH is not often used to fine-tune, but it does have an impact on how the elution looks like. And at the very end, I would just like to share some tricks with you that hopefully make it easier to get started with hydrophobic interaction chromatography. In terms of columns, we find columns with different functional group or ligands on there. So for instance, what Tozo provides are butyl columns, phenyl columns, and ethyl columns that all have a different hydrophobicity and different selectivity. So as a rule of thumb, for less hydrophobic molecules, you need a more hydrophobic column basically. And one thing I would like to mention here is that it's important, not only the group itself determines how hydrophobic the column is at the very end, but also the base matrix and the ligand density. So it comes that, for instance, a butyl column may be less hydrophobic as compared to an ethyl column. It always depends on how many ligands you will have on the column and what the base matrix is. Secondly, we find the columns with different base materials. So there are columns that have a polymeric base material, such as polymethyl. This is what we use, for instance. You find silica-based columns, porous and non-porous columns. Typically, and this is why I always highlighted these, what you will find for protein analysis is butyl groups that have a polymeric base matrix, and that is non-porous. So You would go for a non-porous column if you would like to have low mass transfer resistance and high separation performance. For the porous one, that is more intended for semi-preparative applications, for instance. Also silica, there you are limited with the pH that is used for the separations, for instance. So really the polymeric non-porous particles are the ones that you meanwhile find for HIG applications. I would like to spend one slide on a HIG column that has exactly those characteristics. It's a butyl functionalization. It is based on polymethacrylate, so a polymeric based column, and it is non-porous. That will be new in our portfolio, and it's available in 3.5 centimeter length or 10 centimeter length, and it's really an improvement to what has been on the market recently, as it really shows the highest resolution for antibody drug conjugates and their drug antibody ratios. So we compared this new TSK gel HIG ADC butyl column with other commercial columns, and what we found is that really gives us the best separation of all. So this is something we see if we look into the DAR4 peak over here. We see a small shoulder that we don't see anywhere else. Same is true for DAR6. We have a better separation of our main peak and the pre-peak as compared to all other columns tested, and these included silica particles, polymeric particles, porous, non-porous ones, to basically cover what is out there in the market currently. And apart from the separation performance, we also found that it's very reproducible, which is very important for hydrophobic interaction chromatography, as it is a very sensitive method, in particular to small changes. And we found when injecting 400 times, in this case a MAP antibody, that we also saw the very same profile and retention time over the course of 500 injections onto that column, which is quite a lot. Last but not least, as these columns are stable to high pressure and high flow rates, it's possible to accelerate the analysis, as we can see when using here on the left a column with 3.5 centimeter length only for the determination of drug antibody ratio. We can see that we separate all the different drug antibody ratio molecules, and then we can calculate the drug antibody ratio for this molecule, while the separation only lasts 7 minutes. Now we come to one of the most important parameters for HIG separations, which is the type of salt that is used in the mobile phase. The very typical one is actually ammonium sulfate, but you have the possibility to go from there to other salt types if you see either your molecules are not retarded by the column, or if you have your molecules sticking to the column and cannot dilute them at all. So what you can do in order to have a better binding to the stationary phase and increased retention is to go to more cosmotropic salts, for instance changing from ammonium sulfate to ammonium citrate. On the other hand, if you have the problem of molecules sticking to your column or a low recovery, you may rather want to go to chaotropic salts, such as changing from sodium sulfate to sodium chloride, for instance. An example of how the salt type influences the separation by hydrophobic interaction chromatography, we have here a separation that was done with standard ammonium sulfate, and a monoclonal antibody was separated in this case, and then the same monoclonal antibody separated in sodium chloride. What we see is basically, well, we can, if we look very closely, imagine that we have some fragments over here, but in the later eluting part, which would be aggregates, for instance, there we cannot see anything at all. However, if we look into the sodium chloride separation, or the separation in a sodium chloride buffer and gradient, what we see is separation of fragments and also aggregates. So in this case, we used another salt and thereby changed the selectivity. Now, apart from the question which salt we use, another question is how much of it should we use? This is somehow intertwined because some salts need less concentration and some salts need higher concentration. So if we talk about more chromatographic salts, such as ammonium sulfate, for instance, we need a lower concentration of it. So very typical numbers you can start with is, for instance, for a monoclonal antibody, you could start with 1 molar of ammonium sulfate and go back to 0 molar, so no salt at all, in your gradient. Now, rather chaotropic salts like sodium chloride, for instance, they require higher concentration. So for sodium chloride, if you would like to use it, you would go as high as 3 molar. And what impact the concentration of salt has been shown here in a publication that came out in 2021, where a separation was done on a monoclonal antibody and the salt concentration was increased. What you see is that the higher the salt concentration, the more retention and the later retention times you will get. So this means if you would like to have retention at all, or later retention, or more retention, you need to increase your salt concentration. One last thing I would like to mention is it also depends a little on how big your proteins are. So if you have smaller protein, you would always need more salt as compared to a larger protein. Whereas the salt concentration and also the salt types are parameters that are often used to optimize the method, we now come to the pH that is less often varied in order to get a better separation. But I still want to say some words about the pH and how it influences our Higgs separations. So as a rule of thumb, what we typically do is we start at the pH that is the isoelectric point of our protein, which means our protein will be there in a non-charged form. Now, when does it make sense to actually look at the pH and when it makes sense to change it? This is something that we see here, where we have a mix of different analytes that elute at different retention times when changing the pH. And what you notice is that there are some pHs where all of the analytes basically elute at the very same time, whereas there are other pHs where we see that the retention time is differently. So actually what we get is separation. So in those cases it makes sense to also look at the pH of analysis. And with that, we already landed at some general tips and tricks you can use in order to make sure your hydrophobic interaction chromatography method works. And one thing you should keep in mind is that you use similar salt molarity in the buffer and in particular also in your sample. Because what happens if you have a lower molarity in your sample, just use the normal buffer that it's dissolved in already, you will have a plug where your low salt concentration is in and your hydrophobic parts on the molecules won't be accessible because there's simply no salt or not enough salt in there, and they won't be retained. And this is something that was also shown before in a publication. If you don't have any salt in your sample buffer, then you have a lot of your molecule eluting directly in the dead volume, so no retention at all. When you then increase the molarity in your buffer, of course, you will also get the retention that you would actually like to see. Something we haven't been talking about is the gradient actually. So as a rule of thumb, what you can use to start off with is a linear gradient from your high salt concentration, may it be 1.5 molar ammonium sulfate or 2 molar sodium chloride down to 0 salt, and this linearly over 10 column volumes. Next, we talked about the type of salt that you can use in your separations. What I did not mention yet is that if you have the time to test a mix of several salts, this may also have an impact, a positive impact on your selectivity. So you can tune the selectivity, but it's a lot of try and error in there. Of course, we are using high salt concentrations, so it's important to also have a look at your HPLC system. So you should regularly flush it and clean it when working with such high salt concentrations. And one thing you should also keep in mind that you don't want any of salt crystals or precipitates to block your system, so it really makes sense to filtrate your buffer before attaching it to the system. Last but not least, this is something I kind of hint to already in the columns part. So the use of very small particles and non-porous materials is actually very beneficial in order to get the best separation performance, and this is something you could use, for instance, the TSK gel butyl NPR column 4. With that, I would already like to summarize what we learned today and come back to the title of today's talk: Hydrophobic interaction chromatography for protein analysis. What is it? So we separate our proteins by their hydrophobicity, and we do so by using a salt gradient. Why would we like to do that? Because we can determine a variety of protein modifications by that method, and how we do so is by developing methods where we actually mainly look at the salt type and salt concentration that we modify in order to get the best separation of our molecules.